Well, I'm very honored to be asked to come to speak to you today. It's a delight to be here with so many important microbiologists, particularly from Spain. And I wouldn't miss this opportunity to honor Pepe's legacy. I'm going to come back to the, I think I get that right, that's right. I'm going to come back to the European Academy towards the end of the talk. But I wanted to sort of take you through my connections with Pepe and explain how lots of the things we did together happened. Okay, that goes that way. Okay, got it. So I think you could say the story of my connection with Pepe started on the 25th of October 2001 with the publication of the Salmonella genome sequence. This was a lovely piece of work from Michael McClellan's group in the States. And he was kind enough to share the sequences a couple of years in advance to people who wanted to get in touch with him by the rudimentary methods we had at the time. And this was a real game-changing moment. The first ever bacterial genome was 1995. So 6 years later, we got the Salmonella genome, even though it was from a non-pathogenic strain. So it's very beautiful, as I'm sure you'll agree. And we can understand a lot by looking at the sequence of the genes and trying to work out what they do based on homology, etc. But really, a genome is not going to explain function in itself. And I would argue that really, to be functional, a gene must be transcribed. And I know many of you are fascinated by the regulation of gene expression in bacteria. Now, in the 1990s, and we've heard quite a bit of work going back to the 1990s today, I think many of you were much better at it than I was. LAC-Z assays, beta-galactosidase assays was the way you looked at the activity of gene fusions. Now, here's my lab book. This is what we used to do before we had laptops. We used to write down numbers in a book. The replicates were not very good, and the numbers varied day on day. The numbers would vary, and they allow you to look at one promoter at a time. It was so frustrating when you want to look at many different promoters. So we were lucky that Pat Brown and Joe DeRisi at Stanford developed microarray technology. This shows a robot that we built in my lab in Norwich, which is doing PCR. So we PCR'd up all of the genes in Salmonella typhimurium and Salmonella enteritidis to make a microarray using this printing robot, which we also built to a recipe, a design from Pat Brown and Joe DeRisi. Joe DeRisi designed the software. And what it would do is put spots of DNA of these 5,000 odd PCR products onto a glass slide. And you end up with something that looks like this. This was called the SALSA microarray, standing for Salmonella cerevisiae, which we made in my lab starting in 2001, and then going on to 2008, which I'll come to. It's very pretty. And by you're putting on cDNA with either red or green, you can look at differential expression of the two. So it was really good for looking at gene expression. But microarrays are very dull, and the only really interesting thing is to come up with great ideas to use them. So at the same time as developing the technology, I applied for a grant to the EU. And may I apologize for my government's ridiculous policy, which embarrasses me completely. And without the EU, I probably wouldn't be here speaking to you, because this is how I've formed lots of connections. We got funding for a Marie Curie training site based on microarrays. And this was funded in 2001, called BACFUN. It's got to be an acronym for Bacterial Functional Genomics. And so this was intended to bring PhD students from some of the most interesting groups in Europe to my lab to do some cool experiments with microarrays. What are you going to look at? What sort of environments? Well, we know that during infection, Salmonella is exposed to many different environments, and there are going to be rapid changes in all sorts of things. We can talk about temperature, we can talk about pH, as we've heard about, osmolarity, nutrient availability. All sorts of things are happening during the infection process. And we know that to survive, bacteria have to be fit. And if you're going to make things like these type 3 secretion nanomachines in the outer membrane, you've got to be only expressing those genes at the right time. So to optimize fitness and so cause disease, the salmonella have to express the right genes in the right place at the right time. So what we had to do is understand what they are. So we had to look at every salmonella transcript, and we had to use infection-relevant conditions. So some of those can be in vitro. We could have environment media that mimic aspects of the phagosome, and media in which SPY1 is expressed at high levels. But also we have to look inside mammalian cells and look at real infections. And so one thing that the Marie Curie training site did was bring a PhD student from Mikael Wren's lab in Stockholm to my lab, and Sophia Eriksson was able to develop a protocol to get RNA from salmonella within macrophages. And so we could see what the bacteria were doing during a real infection, which was a game changer for us. And so that took me to the Salmonella Conference, my first ever Salmonella Conference. And this research was inspired by this talk. I wanted to find out when I first met Pepe. So I first met Pepe in Sardinia at this conference, September 2003. Thankfully, it was towards the end of 2003 by the time we published our macrophage paper, because that's why I was invited to speak. But the other person to speak was Pepe. So here was his talk. It was regulation of conjugal transfer and Salmonella virulence plasmid, role of DAM methylation. And what did Pepe's talk focus on? Well, I can tell you because I found my notes. So if you look really carefully, and I can't read it, so I don't know what luck chance you have. Genome-wide search for DAM-regulated genes using a MuDJ screen, of course. Interest in DAM-repressed genes, links with the SOS regulon. Genetic screen for FINP, role in conjugation. And this interesting interaction with the LRP binding proteins and DAM methylation. So that got me interested, and I'm sure that there were conversations after that in the bar, which that built on. Now, so what that led to, after a few glasses and a few chats, is that somebody from Pepe's lab came to my lab, and that person is in the room, Roberto Balbontin. He was the first collaboration with Pepe's group, and I have evidence. This was us in the summer of 2005. This was our really lovely summer's day in Great Yarmouth in Norwich. Now, those of you who look carefully will see the type of coats that we're wearing. It was so cold and rainy. And you'll see Roberto there at the end. So this was my lab at the time. And I'm also revealing my very poor skills at Photoshop, because I actually put the horses in later, and you can see that they're slightly truncated. So I just want to be extremely honest about that. And the result was this paper, Roberto's very nice paper, that came out in JBACT, describing what DAM methylation did in Salmonella in terms of modulating virulence gene expression. And it's been cited reasonably well, which was great. So the microarray was powerful, and it allows us to do quite a lot of things. But of course, it all changed. 2008, new technology came along, and so we moved towards RNA-seq. And RNA-seq was brilliant because our microarray was limited because we only had coding sequences, and only had coding genes on there. We didn't have small RNAs, and of course, being a friend of Jörg Vogel, we had to look at small RNAs as well. We decided to go into this theme of what are the Salmonella doing during infection in a bit more detail and focus on some more environmental conditions, and specifically ones which are happening during infection of the gut and things that are happening during infection of the macrophage. So we came up with about 20 infection-relevant conditions, particularly pH of the stomach or bile. So one of the things I wanted to talk about as I'm giving this presentation is that while we were developing all our ideas, Pepe's influence, I think he used to sit on my left shoulder, was always to say, oh, don't forget about these other interesting environments, Jay, like bile. So bile has always been included. And what concentration of bile do we use? The concentration that Pepe's lab used, naturally. And we were looking at other things, osmolarity, temperature, and then things in relation to life inside the vacuole, in the macrophage, a different pH, reactive oxygen species, reactive nitrogen species, etc. And the way we did it, in case it's of interest, is we did 10-minute shocks, because many of these stressors are really iniquitous to the bacteria and will slow down growth a lot. So if you grow, say, if you try to grow a culture, say, to mid-log, the growth rate in presence of peroxide may be fairly poor. Whereas if you do a 10-minute shock, the growth rate will be about the same. But you can see all of the genes, Salmonella switching on to detoxify that. 10-minute shocks, I think, will be useful for all sorts of different bacterial work. So it proved very useful for us. We were looking, when we look at the expression in all of these 20 conditions, which I'm not going to bore you with, overall about 86% of the Salmonella genes were expressed in at least one of the conditions. And about 8% of those genes would be switched on if the bacteria were in contact with the surface. We didn't add a biofilm condition or an agar plate condition to this. So this made some sense, and we saw that most of our environments, we were getting expression of a majority of Salmonella genes. And one's there, bile shock. So we didn't just do a growth in bile, we added bile and then did took the RNA out a few minutes later. So one of the things I wanted to do in terms of, I've always believed it's really annoying to have to go find a paper, find a supplementary, find an Excel file, get all the numbers, try and understand some really complicated experiment when you've got 5,000 rows and 60 columns. It's not the easiest thing to do. So I resolved to make our data freely accessible, and accessible so you could look at it in the pub. So this database, which is called the SALCOM, Salmonella Compendium, and actually this one I'm highlighting is SALCOM Mac, will work on a smartphone in a pub. And so you can look at expression of your favorite genes in all of the conditions. When you come up with a bizarre idea, you just want to look at. And because everybody hates URLs, if you just Google for SALCOM Mac, the top hit, click on that, and that'll take you straight in. And you can put in a gene of interest, you can put in a gene name like LPX and then an asterisk. That's a wild card, allows you to see all the LPX genes. And you can also go in and look at the actual genome. There's a link to a thing called JBrowse, which is nothing to do with me, which allows you to look at the transcripts in the context of the genome. So please do go and use it. These are some papers we published. This is the compendium, came out in 2013. We did of course, we did the intramacrophage transcriptome with the RNA-seq, so we could look at the small RNAs. That was published in 2015. And also inspired by Pepe, because the conversation basically went something like, well, that's fine, Jay, in looking at the environment, but what about regulatory mutants? So, PhD student, very talented PhD student, Aoife Colgan came to the lab, and she started making some really clean mutants. We had to make each mutant really, really carefully, removing the antibiotic resistance cassette, et cetera, et cetera. Along here, we've got lots of regulatory genes that affect SPY2 gene expression, a lot of regulators that affect SPY1 gene expression, and this one here, DAM. So, of course, we're coming back again. I'm glad DAM has already been introduced, because Pepe was still sitting here, so I had to include his favorite regulator. And just to mention to this audience, there are 3 small RNAs which are downregulated in this experiment. So I'm really hoping somebody might be following up on those. The other thing Pepe is really interested in, hasn't really been talked about much, is plasmids. And this is an interest we share. It seemed that plasmids seemed to be really important for bacterial virulence in the 80s. And then in the 90s, people got so fascinated by the chromosome, they started to ignore plasmids. And that happened in the early 2000s as well. But I was always really keen to make sure we understood what the plasmids were doing in Typhimurium. In our strain, which is the parent of SL1344, you can see 3 plasmids: collagen plasmid, virulence plasmid, a small plasmid. So I had a PhD student who was starting to look at the transcriptome. He made an interesting discovery. This is showing you the primary transcriptional start sites, so the normal promoters in front of genes, which you'd expect. But in fact, in plasmids, there's also a very large number of antisense transcriptional start sites where promoters are going the other direction, which I think is telling you something important about the biology. So when it came to Satish's viva, the only person in the world who could really do this viva properly was Pepe, and I persuaded him to come to Dublin, and here is Pepe with Satish and me and the examiners. This is me with Charlie Dorman and Pepe just before we went for dinner. Pepe wearing a tie, I was very honored by that. He didn't have to wear the stupid black gown you need at Trinity College Dublin, because this was when I was in Dublin. And this is Pepe showing actually how he was with many students. He was so encouraging, so interested in their development. And he did a lovely job with Satish's Viva. And what we ended up with, it was a transcriptome of these plasmids. And you can look at the transcriptome, and you can look at there are some antibiotic resistance genes which are highly transcribed. This is expression on the negative strand. This is expression on the positive strand. So you can look at genes which are being highly expressed in many conditions. And you can see some are called STNCs. These are new small RNA candidates that we found and annotated now on the plasmid. And for those of you who are interested in plasmids, we have actually made this data available. We haven't published it yet because there's a lot of follow-up work going on. But if you go to Salcom D23580, that's the code word, all of the data for the plasmid expression from this 474 strain, this SL1344 parent, is there. So you can see all of your favorite plasmid genes like the SPV genes and the TRA genes when they're expressed, and you can look at them in the context of the chromosome. You can go into the JBrowse and select for the plasmid. I'll be very happy to show anybody who is interested. I really hope this data will be available. And I've never announced before that this data is available, but I just like people to use it even before it's published. But then in 2010, a new disease was revealed, which is causing a lot of death in Africa. So I decided to refocus my lab about 12 years ago on this new type of salmonella, which is causing a lot of mortality, particularly in West Africa, but also Central and Eastern Africa. So I've moved away from this classical type of ST19 salmonella that causes gastroenteritis towards this African salmonella. And I had to mention this because of Stan. And when somebody says something like you did, Stan, I suddenly had to change my talk, so I hope this is going to work. Just to give you the context, about 500,000 people have been killed by salmonella bloodstream infections. These people are largely immunocompromised by HIV or malaria or malnourishment. And 2/3 of it is caused by one type of typhimurium, ST313, that is only found in Africa and has evolved specific characters, which I'll briefly tell you about in a moment. But while we're thinking about what's actually happening in Africa, we are very lucky. We are so lucky in the salmonella community, because salmonella is the most highly genome sequenced bacterium in the world. I think SARS-CoV-2 will come above it. And so you can see the number of salmonella genomes in the SRA compared with all other bacterial species is huge. The up-to-date numbers, which I looked at this morning, this shows you the number of genomes available through Enterobase, a wonderful resource if you're interested in genomes. Was not very much until 2014. And then the new Illumina machines came out, then the number of genomes massively increased, and this happened. And as of today, we're up to 412,000 genomes. So there's a lot of things that could be done with those, particularly with some of the proteins that people in the room are interested in. We understand the taxonomy and evolution of Salmonella genus much, much better. And we found some new subspecies that hadn't been found before. My lab looked in venomous snakes, things like puff adders and black mambas, green mambas, and found lots of them were carrying salmonella, some of the salmonella new to biology, which is in a separate paper you could look at. And yes, I think I won't dwell on that. And then we did our own sequencing because we realized there weren't enough genomes from Africa. So we ran a 10,000 genome project, and we ended up with about more than 10,000 isolates from 53 countries, dating around 68 years. So obviously, the most recent ones are about 2017, 2018. Went back to 1949, and this is our team. This is me with a team of scientists and clinicians in Malawi, where much of this work is done. This was called the 10,000 Salmonella Genomes Project. And the result of it was we started to understand how the evolution of ST313, this type of typhimurium in Africa, has been going along in a stepwise a stepwise way that could have been predicted by what Stan said earlier. So this is what's called a chronograph, a Bayesian, a beast analysis of 150 genomes. On the horizontal axis is time, and vertically here we can see different lineages of Salmonella typhimurium. This is ST19, which is normally responsible for gastroenteritis. We have a lineage 1, which has largely been replaced now by lineage 2 in Africa. And so lineage 2 is what I want you to focus on. And if we ask what's been happening, this is what's been happening. Many mutations have been coming at different times, and I'm not going to go into the details, but these are pseudogene mutations. So Stan, you're completely right. It's largely about pseudogenes. So we see the African Salmonella as starting to specialize. It's adapting, but it hasn't yet become a specialized organism. It could still infect other animals at the moment. So I can't go into these in detail, but these are the key genes, some involving catalase, melibuyers metabolism, SSEI, an important effector, is inactive in the African Salmonella. And if they're inactive, they're all pink here. And you can see that these mutations have been fixed in the lineage. This is telling me it's not just random, but it actually gives those bacteria some competitive advantage. And for those of you who've heard about our story involving the PGTE virulence factor, we found one nucleotide responsible for upregulation of that in African Salmonella, which I'm afraid you'll have to read about elsewhere. And these red ones are carrying this single mutation. So in all of the strains we were looking at, we found this one T nucleotide, which is, now we've seen that in thousands more. And this was published a couple of years ago, and Casey Pulford led this work. So next I ended up in Oslo, as you do, Genome Maintenance Conference, where they wanted me to talk about some of the approaches we were using. And guess who was there? Pepe, because Pepe's interest in DNA repair took him there, I think. We had a wonderful dinner at the Oslo Opera House, and then this was taken in the City Hall, where they give the Nobel Peace Prize every year, and Pepe really enjoyed telling me about that. And then this is the last photo I have with Pepe, where we're in reality. Do you know where it is? Any guesses in the room where this might be? Train station. Very good. Very good. So this is Seville rail station after an RNA meeting that I was at, and then I had a meeting with Pepe and some people from his group. And so the next stage in our collaboration will be this paper, which I'm supposing will be our last collaborative paper. This was driven by work that Ankla did when she came to my lab in 2017. And we have a tradition in my lab that you need to wear clothes carrying bacteria which are of interest in your research. And these are some of Ankla's GFP-expressing Salmonella, of course. And now we're working with Francine, doing some of the data analysis. And this paper, I'm hoping, is going to be informative and interesting. And that was the last interaction I had with Pepe, which was on Zoom in mid-22. The results are going to be lots of transcriptomes comparing the two main strains, 14028 and SL1344, see how similar and different they are. And this is Pepe. Now, where's the mouse? The mouse over here. How do I turn on this? I might be able to do it here. This is Pepe giving us a little bit of his wisdom.
I remember what you said when we were traveling on the bus. So it's true that, I mean, the circular representation can be misleading.
And he's right. And yesterday I was talking with Francine about the circular representation, and we've now come up with a better way of doing it. So thank you, Pepe.
I remember.
So I've done that. And now I talk about the Academy. Okay, the European Academy of Microbiology is a grouping of microbiologists set up about 14 years ago to promote excellence in microbiology across Europe. Members of the Academy are experts in microbiology and have either had interesting papers or interesting patents or interesting inventions in this field. There are 160 elected fellows from 26 countries. We've had some illustrious presidents, and actually there's been an election. We are going to have a new Secretary General next year. We have a board which helps to manage the organization. which I'm part of, and this shows you who's involved, including Victor, of course, very important member and very active in the academy. The fellows come from all sorts of countries, shown here. And one thing I noticed looking at this graph is there are not enough people from Spain. So this is something, perhaps by coming here and talking a bit about the academy, it might at least explain to you what it is. I think most people have never heard of this grouping of senior microbiologists, partly because we have failed to make this a really visible and interactive community. If you want to go and find out some more details, you can go and find this members book, which will talk about each of the people who is in the academy. And Pepe has his own page. And he was elected as a fellow of the European Academy because of his contributions in the area of genetics, salmonella gene regulation, epigenetics, and transcriptional control via DNA methylation. So at that point, I'm going to leave you with a photo of Pepe at the European Academy of Microbiology retreat in Segovia in April last year. And you might notice Victor's standing up here, and there's Danny Lopez. Oops. So I think that now tells us we have to go to lunch. But apparently there's a photograph first. So let's do that first. Thank you.